Come on and cry me a river, cry me a river. I cried a river over you.
Hello everyone and welcome again to this day. What a day, right? Very important day, a lot of changing, so welcome. Not necessarily talking about politics, but at the end of this emission we may visit a new website I found that is worth monitoring, so we can monitor the situation together for a little bit, just to try the tool, a very interesting tool. This is the one I'm talking about. Take a look. Okay, this is a link, of course. No, this is the GitHub, and here we have monitoring the situation, the link. But we're gonna wait for that. It's very cool visual overview of the world news. Then today I wanna read with you this thread or article or post about Neuralink and its new patent. We still don't understand human cell behavior. That's about to change. An article from Big Think. Oh, it doesn't show. There you have it, Big Think. And an article from John Templeton Foundation. Plato warned us about ChatGPT and told us what to do about it. I don't know exactly what's about, so we're gonna discover it together today. Saturday normally is a very slow content day, but today with the news of the world, it's a little bit not a little bit, a lot more move today. So we're gonna do what we're gonna do. Let's start with one. With which one? This one. Or this one? I think this. Plato warned us about ChatGPT and told us what to do about it. We have the chat. Last time I forgot to read the chat. Our first two chats and I missed it. Sorry. I'm ready today to read it because it was too small in the monitor and I was very concentrated reading because it's hard to me to read in English. Very hard. So let's go. I'm gonna leave the links for the persons who just wanna read by themselves. Nice. Long before the printing press or ChatGPT. The music is too loud. Okay. Here we go. Long before the printing press or ChatGPT, Plato was already worried about what new technologies might do to us. In the Phaedrus, Socrates explains why he never wrote anything down. He tells us that when writing was invented, its inventor boasted that it would make people wiser and help them remember. But in his view, it would do the opposite. It would make people forgetful and give only the appearance of wisdom, not its reality. Okay. Written words are prone to misinterpretation. Yes, like chat. They are indifferent to their audience. They are a kind of dead speech. Wow. As a philosopher of science and AI who also teaches ancient Greek philosophy, I'm struck by how closely our current situation mirrors the concern of the ancients. LLMs like ChatGPT are new technologies that promise to make us wiser, to read, write, and think on our behalf. But like writing in Plato's time, they threaten to erode the skills they imitate. In a recent Templeton Ideas essay, Richard Lopez articulated 3 perceived harms. AI gives us quick answers, but robs us of understanding. It displaces real human interaction, and it stunts personal growth. Many others are sounding the alarm and calling for tech-free spaces. But would Plato agree? The irony is that we know Socrates' warnings about writing only because Plato wrote it down. Did he forget his own criticism or decide to trade off was worth it? That's a good plot twist. That's correct. When I teach the Phaedrus, I follow my former philosophy professor at Boston University, David Ruchnick, who argues that Plato was well aware of the force of his own critique of writing, but believed his work could overcome its limits. Plato wrote dialogues because they could come alive in the reader's mind. They invite reflection, questioning, and response. Such living speech enhances the reader's faculties and even discriminates among readers, refusing some and rewarding others. Socrates, for example, often gives arguments that are mediocre. This is deliberate, some will be satisfied and move on, while the more philosophical-minded will object and dig deeper. In that sense, every Platonic dialogue is 2 conversations, the one Plato wrote and the one that unfolds between Plato and the reader. We have a quote here. If you're looking for a call to action, I suggest we take the lead from Plato. In the age of writing machines, we must reinvent writing itself. Ooh, that sounds good. Any ideas? Okay, crisis in the classroom. How might we do that? Reflecting on Plato's model of education in the Republic is a painful and difficult turning of the soul. The arrival of AI that can write and think for us is oddly invigorating. It invites us to reconsider writing, not just as a tool of communication, but as an art and practice of thought itself. Who's the author? Eli Schick. Cool. Shout out to Li Chic. Where was I? Okay, I don't know. But would Plato agree? The irony is that we know Socrates warning. That's all. How might we do that? Reflecting on Plato's model of education in the Republic is a painful and difficult turning of the soul. The arrival of AI that can write and think for us is oddly invigorating. LLM technology and its wide accessibility push us to ask what understanding, creativity, and authorship truly are. Can I understand something if I can't say it in my own words? Or if AI can say it better? If the idea is mine but the text was created or published with AI, am I still the author? There is no doubt unreflective LLM use might lull us into complacency. But the questions raised by these systems are vital. For Plato, such intellectual provocation is the beginning of wisdom, like the prisoners in the allegory of the cave, turning from shadows toward the light. We are being forced to turn from taking writing for granted to questioning its value in our lives. That turning can begin anywhere. We might build AI systems that optimize the truthfulness, are explicit about their reasoning, and reveal their biases. In everyday life, we could foster forms of literacy that teach us how to understand the new algorithm shaping our world. Yeah, like the new lexicon from Luke Maxing. And in the classroom, AI can become a kind of living speech, a teacher, editor, and interlocutor. As a teacher, for instance, I can encourage my students to treat LLMs like a Socratic partner, to challenge and test their views, to help them discover what they don't know and expose blind spots, and to use AI in what Angel Scholars calls an exploratory battle, to discover inquisitive opportunities. And since even the best teachers have limited time with students, we can show them how to use an LLM as a personal line editor. Not to replace their writing, but to offer sentence-by-sentence suggestions that help them become better writers. Of course, such techniques are no substitute for human interaction, but they do address Lopez's earlier concerns about substituting quick answers for real understanding and stunning intellectual growth. Still, beneath these practical strategies lies the deeper issue, or a deeper issue. Across academia, from lecture halls to the editorial boards and university committees on which I serve, the same questions resonate. What should we still teach our students, and what standards should guide us? Guide our own writing? What exactly is writing for? What writing is for? Familiar answers come readily to mind. Writing matters because it's how we learn and nurture reflection, distinguish between strong and weak prose, bring human individuality, culture, and taste into place, practice the aesthetic craft, and so on. Personally, I often don't know what I think about a topic before I sit down to write about it. But perhaps that's a far cry from claiming writing is indispensable for thought. There are different forms of thinking and knowing. My own view, echoed by friends and colleagues, is that writing plays a special role in cultivating philosophical thinking, a kind of dialectical literacy that we all need. To see what I mean by dialectical literacy, let's look at what good reasoning is all about. In some cases, all that matters is logical form. Take the syllogism, all Greeks are mortal. Socrates was a Greek, so Socrates was mortal. We could substitute any content as long as the structure holds. But most real arguments live in Messier territory. Sounds like a French word, Monsieur. In Messier territory. These are what philosophers call inductive or ampliative arguments. They hinge on word choice, background facts, and specific content, what is actually said and whether it's true, not just the logical form. That's reading and writing work all the way down. Reinventing writing. Consider then how LLMs increasingly present chains of reasoning, not just answers. When you are dealing with a real-world reasoning, evaluating those chains can be cleanly separated from reading and writing, and yet this is precisely where LLMs can shine if used in the right way. You can literally interrogate them, ask for reasons, demand sources, and evaluate their arguments. It's much more like a Platonic dialogue than static writing that cannot answer back. Yes, that's true and interesting. Recall, for instance, Socrates' worry about how writing is a one-way communication that doesn't adapt to its readership. Nigel Warburton argues that with digital technologies such as online chat, written communication is now interactive, thereby overcoming these limitations. Even more so, I would add, in the era of LLMs. No chat? Am I missing a chat? No. Cool. Here I have the window. And you have it as well. In the left corner of your screen. Down left. Let's continue. We might even say that, like Socrates in the Phaedrus, LLMs let us take refuge in discussions and investigate the truth of things by the means of words. In other words, today's AI systems make the dialectical practice cherished by philosophers readily available. Cool. I like that perspective. How's the music? Too loud? This is no substitute for Socratic conversation in the classroom, but it does suggest teaching, writing, and doing research in a way that can complement this practice. LLMs can function as a real empowerment. The challenge is to use them in ways that deepen rather than diminish our thinking. Okay, we have two chats here, but they are just ad. Hey, I checked out your videos and noticed you're still new or geek. Yeah, they want to sell me views or something like that. No, thanks. I work the other way around. Where I was. For example, just as LLMs can write our sentences and AI apps like Suno can compose songs, before long, a robot may even play all our instruments. But a musician doesn't stop playing just because a machine can make the notes. The point is the playing. So too with writing. Its worth isn't just in the product, but in the process, in what it draws out of us. So, once more, why does writing matters? The suggestion here is that it's essential for the kind of philosophical thinking and dialectical literacy that I described. In short, good reasoning. That it's an art with its own intrinsic value. And how can LLMs function as empowerments rather than substitutes for human thinking? We can use them to broaden our research reach, refine our writing thought, personal editing, and engage in Socratic dialogue, a sustained thoughtful exchange of prompts and responses that becomes a new genre of writing, an AI dialogue akin to Platonic dialogues. Cool. Nonetheless, if we look to this essay for the answer or to anything specific that Socrates says, we miss the point. Plato wrote the dialogues to draw readers into thinking for themselves. Likewise, the task is ours alone and in community, and in community, to decide why writing matters to us and where it doesn't. That's harder, and it's work we can't it's a work we cannot outsource. Okay, Elias Sech is a professor at Auburn University who writes about science and philosophy. His book includes Idealization in Physics and the Metaphysics of Color, and his articles appear in outlets such as the New York Times and the Washington Post. Thank you very much, Mr. Elia, or Elia? I don't know. This is one. Very illustrative. What should do next? What should we do next? Let me see. Let's go to this one. Neuralink has engineered an artificial cortical layer called Layer 0 to serve as a living biological bridge between the human brain and computer. Wow! Finally here. A quick summary of the living brain interface patent. It's a new patent that is out there from Neuralink. Neuralink has engineered an artificial cortical layer called Layer 0 to serve as a living biological bridge between the human brain and computers. Okay, let's gonna fast track the Platonic dialogue with AI. Traditional rigid implants act like foreign objects that damage the brain and trigger scar tissue, which ruins the electrical signal over time. By growing a custom layer of living brain cells directly on the surface, Neuralink avoids this damage completely. Once placed on the brain, this new layer of cells mature much like a developing mind. Wow! Can you believe this? It's a patent now. It transitions from synchronized chemical bursts into the natural, independent rhythms of a healthy brain. Instead of fighting the new cells, the natural immune system actually acts as a supportive caretaker to help them grow and connect seamlessly with the existing tissue. These engineered cells act as a biological translators that can both read natural brain activity and send commands back into the mind. In live testing, subjects quickly learned to consciously control this living interface to perform tasks and earn rewards. This proves that a biological graft can function as a highly precise, two-way communication hub. Before this reaches human patients, engineers must figure out how to grow these interfaces using the personalized cells of a patient to prevent severe immune rejection. Okay. From where they're gonna take this material? From which part of our body, I mean? Oh no. Ultimately, this biological approach promises a permanent, lasting connection that could power future medical breakthroughs. Advanced Tesla robotics and SpaceX off-world exploration that fully appreciate how this biological marvel functions. We need to explore the exact science and engineering that makes it possible. Let's go! Let us dive into the full details of how this organic interface actually comes to life. The dawn of biological interface. The boundary between biological intelligence and artificial computing is about to disappear completely. For years, scientists have struggled to solve the problem of brain implants causing progressive tissue damage. Neuralink has developed an elegant workaround by abandoning rigid hardware in favor of a biological neural layer. This new interface involves pouring a special formulated hydrogel packet with living cells right onto the surface of the brain. Wow! Once settled, these engineered neurons naturally reach down into the existing cortex. They write themselves directly into the communication network of the brain, just like natural tissue. The result is a bidirectional communication hub that reads and writes data without causing a single microscopic tear in the brain tissue by utilizing the very same biological building blocks as the human mind. This invention paves the way for a truly frictionless integration between our thoughts and external device. Just a little bit of gel or your brain. To understand why this radical biological shift is so necessary, we first have to look at the severe limitations of current hardware. The problem penetrating electrodes cause progressive tissue damage. Wow. Traditional brain-machine interfaces rely on inserting rigid probes directly into the fragile tissue of the brain. Think of this process like driving a stiff nail into the delicate sponge. Each penetration inevitably displaces surrounding tissue and disrupts the delicate microvasculature. This is the complex network of tiny microscopic blood vessels that keeps the brain nourished and healthy. This physical trauma immediately triggers the natural immune response of the body. The biological system quickly activates neuroimmune cells, acting as the microscopic security guards of the brain to address the damage. These defender cells work to isolate the invader by encapsulating the foreign object in the dense glial scar. This essentially builds a thick biological wall around the device. Over time, this fibrotic encapsulation acts like heavy cellular insulation that blocks electrical communication. This build-up severely degrades the signal quality and limits the overall longevity of the implant. Engineers have naturally tried to soften the blow to the brain, while newer flexible probes help reduce the severe biological rejection. The sheer physical displacement of the tissue remains a significant barrier to scaling up this system. Even a flexible wire takes up physical space. Recording from millions of neurons simultaneously would require shoving millions of wire into an already tightly packed organ. This spatial limitation makes it clear that gathering massive amounts of neural data requires entirely new methods that do not rely on traumatic physical penetration. This exact realization is what drove Neuralink to develop an entirely organic alternative. Is this true? I'm starting to doubt. Too amazing, because they have the machines to do the implants of these hard chips now. They have patients already. I don't know. Let's see. Do we have any chat? No, we're okay. Neuralink's solution: grafting a living layer of neurons directly onto the brain surface. To bypass the limitation of rigid hardware, Neuralink proposes creating a biological bridge known as a layer 0. This involves transplanting a layer of genetically modified neuronal cells. Genetically modified, wow, which are brain cells specifically customized in a laboratory, directly onto the outermost surface of the cerebral cortex. Wow, specialized and customized in a laboratory. This target area is the heavily wrinkled outer layer of the brain, responsible for higher level functions like thought and action. Once deposited, these customized foreign cells actively grow and integrate into the host brain. They extend tiny dendrites and axons, functioning essentially as the incoming sensory branches and outgoing communication wires of a cell, downward into the natural cortical layers. By doing this, they form actual chemical synapses with the existing neural network of the patient. Wow! I wanna see an animation video of this. I'm gonna ask Grok. These synapses serve as the biological communication junctions where cells pass messages to one another using natural chemical signals. Because the interface is made of living tissue rather than metal or silicon, the brain does not recognize it as an invading foreign body. This natural integration allows the brain to welcome the new cells like friendly neighbors. It effectively blurs the line between the biological host and the technological implant. The result is a seamless connection that completely avoids the problematic scar tissue formation that plagues traditional implants. Watching this biological integration unfold over time reveals a highly dynamic and orchestrated process. Okay, let's see the timeline. The developmental timeline: maturing the graft through synchronous waves. Upon transplantation, layer 0 does not immediately function like a mature brain interface. The patent reveals that the graft, which is the newly transplanted layer of living tissue, undergoes a fascinating developmental process over several weeks. Ooh, let's see. This growth period closely mirrors natural brain development in a growing organism. During the first few days, the cells exhibit large and infrequent synchronous population waves of calcium activity. Oh no! They show cortisol spikes. You can think of this calcium activity as the chemical spark that ultimately powers the electrical signals of the brain. These synchronous waves mean that the cells are firing all together in unison, much like a choir singing in a single, booming note. Ooh! These early synchronized firing acts. This early synchronized firing acts as a biological heartbeat and is crucial for establishing initial network connectivity within the new biological layer. As time passes, the behavior of the cells becomes much more complex. By the fourth week, this massive synchronized firing gradually decorrelates and sparsifies. This means the cells stop shouting all at once. And begin having independent, localized conversations. Cool! The neural activity transitions into asynchronous patterns where individual cells fire on their own unique schedules. These independent rhythms perfectly mimic the natural, spontaneous electrical chatter of the native cerebral cortex, which is the original host brain tissue. This successful mimicry proves that graft matures into healthy brain tissue. Once the cells mature and synchronize with the host rhythm, they begin forging physical connections to prove they are truly communicating. Foreign physical connections, cellular integration, validating true synaptic connections. Who's the author? I forgot to. This is from T.S. Laming or Ming. Great. Cellular integration. Creating a physical layer of cells is just the first step, and proving it actually talks to the brain requires precise histological evidence. Histological is from tissues, if I'm not wrong. This means gathering undeniable proof through the detailed microscopic study of biological tissue structure. The patent details high-resolution confocal imaging, which is an advanced laser microscopic technique that captures incredibly sharp and three-dimensional views of the cell. This method was used by researchers to clearly trace the exact connection between the implant and the host. Researchers observed that the transplanted neurons grow mature dendritic spines. These tiny thorn-like protrusions act as the primary signal receivers for neural input, and they reach down deep into the superficial layers of the host brain. These spines possess specific postsynaptic markers such as ZINGAP1. These markers are specialized proteins found on the receiving end of a neural connection, and they act as biological docking stations to indicate the presence of active communication hubs. These structures closely align with presynaptic markers, which are the matching proteins located on the sending ends of the native tissue. This exact alignment proves that genuine excitatory chemical synapses are actively formed. These specific synapses are the vital communication bridges that stimulate neighboring cells to fire and pass along information. The graft even receives inhibitory input from the host, which consists of calming signals that tell the new cells to quiet down and stop firing. This demonstrates that the engineered layer fully integrates into the delicate electrical balance of the brain. Achieving this level of seamless integration is only possible because of an unexpected ally within the host. Is because an unexpected ally within the host. The immune response flipping the host defense mechanism. Typically, the immune system of the brain actively attracts and walls off foreign implants to rapidly reject unfamiliar objects. Neuralink flips this defensive response completely on its head with their biological approach. The patent notes that host astrocytes and non-phagocytes microglia actively migrate into the transplanted graft. Astrocytes are star-shaped cells that function as the master architects and caretakers of the brain. Oh, I didn't know that. Meanwhile, no phagocytic microglia are specialized immune cells that have shifted from an aggressive, cell-eating mode into a calm, supportive surveillance state. Rather than attacking the new cells, these native immune cells act as caretakers and provide essential metabolic and structural support. Okay, what is this? Aikido in a cellular level. This means they supply vital energy and build a physical scaffolding to keep the new tissue healthy. The host astrocytes even help seal the biological layer to the skull and encourage new blood vessels to grow directly into the graft. Wow, unbelievable. This natural vascularization is the process of building a biological plumbing system to deliver constant oxygen and nutrients. This represents a massive technical victory, ensuring the long-term survival and health of the interface. With a healthy, vascularized graft securely established, the system is fully prepped for the ultimate goal of reading and writing data. Wow. 4 weeks? Optical and electrical communication, reading and writing neural data. The transplant cells in layer 0 are carefully engineered to act as a natural transducers. A transducer, this is what I know about the term, is a device that transforms one kind of energy to another kind of energy, like the microphone transforms air pressure to electric signal, right? And the speaker makes the opposite in the other direction. From electric signal to air pressure in the speaker cone. So, the transplanted cells in layer 0 are carefully engineered to act as a natural transducers. They function as biological translators that can convert internal brain activity into readable formats. One approach involves using genetically encoded calcium indicators. These are specialized proteins acting like microscopic, built-in flashlights that cause the cells to emit fluorescent light when they fire action potentials. These potentials are the sudden electrical spikes neurons use to communicate with one another. A small optical window placed over the skull allows a tiny microscope to read these light signals in real time. What? A small optical window? Placed over the skull allows a tiny microscope to read these light signals in real time. Alternatively, these cells can be chemically tethered, essentially bound using specialized molecular Velcro. What? To an ultra-thin surface electrode array. Okay, I'm lost here, but let's continue. Or not. Maybe not, it's too long. Too technical. What do you say? Yeah, it's very long. Let's go to the next one. I'm gonna leave the link below in the video in YouTube so you can read it complete. But it's too hard, too technical, right? How's the chat? Oh, only promotions. What do we have? Let's monitor the situation for a little while so that I can test my computer if it can do that or not. Maybe I crash, so I'm sorry if my computer crashes. I'm gonna check out. Let's monitor the situation. I don't know, it's too heavy. Too heavy for my MacBook Air from 7 years ago. Okay, there we have it. Monitoring the situation. We have live cameras. Oh, the music, it's done. Let's go. I'm gonna mute myself for a little bit while I navigate this portal. Oh no, my music is crashing too. It's too heavy. Okay, it's too slow. I'm trying to move things, see, check, but it's too heavy for my computer to do all these things at once. So I'm sorry about that. And what else we have here? I'm gonna log out this heavy. Let's read this one, and after that, I think it's a good, the most, no, it's not the most, it's the longest life I made until now is today. So let's do this. We still don't understand human cell behavior. That's about to change. By treating the human body as an information system, scientists are using AI to simulate cells, visualize hidden biology, and detect disease at its earliest and most preventable stages. By Lee Hilland, February 18, 2026. This was created this is an article created in partnership with BioHub. Okay. Good to know. The human body is a problem. For most of us, it's a management problem: nutrition, exercise, blood work, the occasional X-ray or MRI, and treatment regime. For doctors, it's a disease problem. What drives the formation of amyloid plaques in Alzheimer's? Which factors make certain cancers resistant to therapies? Why can't we detect Parkinson's sooner? Doctors trying to study these questions may also say the human body is a founding problem. And for researchers in the age of artificial intelligence, the human body is a math problem. There are on the order of billions of proteins, just as an individual human cell. The 3 billion nucleotides in the genome specify the entire program for the human body. 3 billion nucleotides in our genome for the human body. There's just tremendous complexity, says Alex Rives, PhD. To the 400th power possible mutational patterns that can lead to cancer, he says. It's more than the number of atoms in the universe. How is that possible? We spoke with 4 BioHub scientists working at the frontier of this challenge, using the new tools, new data, and AI to make the body's complexity legible. The missing science of cell behavior. Over the past few decades, scientists have mapped the human genome, identified thousands of cell types, and learned to image living tissue in remarkable detail. Yet a basic question remains unanswered. How do human cells actually behave, moment to moment, under stress in relation to one another? And how do those behaviors tip the body from health into illness? In cancer, researchers can identify many of the mutations linked to tumors, but still struggle to predict which altered cells will turn deadly. 2 patients with nearly identical tumors can respond very differently to the same treatment, often because of differences of how cells signal, adapt, and interact with their environment in ways we can't yet reliably observe or control. In Alzheimer's and Parkinson's, pathological proteins accumulate for years or decades before symptoms appear. But the tipping point, the moment at which a patient begins to show signs of dementia or disability, remains poorly understood. By the time memory loss or movement problems emerge, irreversible damage has already occurred, leaving clinicians with few options beyond slowing decline. Simulating life at the cellular level. Because scientists can't yet observe living human cells as disease unfolds or rewind to the moment something goes wrong at the molecular level, they're turning to AI to simulate cellular behaviors across time and conditions that are difficult to capture in the lab. You can think of the human cell as a system of information, says Rives, a pioneer in AI and biology who developed the first large transformer language models for proteins. Biology is a kind of language. What would it mean to train a model on the text of life? Wow. Using massive datasets and scaling up to 10,000 GPUs of compute over the next few years, Rive's BioHub team is building virtual cells, AI models designed to behave like real human cells, much as a language models can behave like a therapist or a math tutor. Cells are made up of molecules. They come together in complex circuits. All of this is programmed by the genome. There's many different layers, Reeves says. The complexity is far beyond the ability to simulate from physical or chemical first principles. You need an information theoretic-centered approach. As a step toward building virtual cells, BioHub is scaling up the generation of data. Its Billion Cells project is an early effort to map the many states a human cell can occupy. Rives believes that training AI on such a map could make it possible to model the underlying mechanisms that govern health and disease. BioHub's technology development efforts across frontier imaging, instrumentation, and measurement are expected to expand both the breadth and resolution of that map over time, giving models a more detailed picture of human biology. Ultimately, Rives' team aims to leverage AI to speed the search for ways to prevent and cure disease. From snapshots to imaging systems. Imagine trying to understand the inner workings of a city from a single aerial photograph. You'd see buildings in one moment of the water, but not traffic patterns, the power grid, or how the environment changed over time. Modern imaging tools such as X-rays, CT scans, and MRIs work much the same way. Invaluable for spotting tumors, blockages, and structural damage, but usually only after disease has taken hold. Events that contribute to disease can happen at the molecular level in milliseconds, says Scott Fraser's PhD, president of imaging at BioHub. In type 2 diabetes, for example, pancreatic cells can compensate for dysfunction for years before blood sugar levels rise. To see the early signs of trouble, we have to be able to scale from molecules to whole bodies, a billion-fold change in scale, and almost the same change in time. Wow! Further, even the most advanced microscopes leave much of biology unseen. If we ask what fraction of the proteome, the full set of proteins active inside a cell, we can really look at, it's probably less than 1%. So less than 1%, Fraser says. By combining multimodal imaging with AI models that guide both data collection and image interpretation, Biohub is expanding what researchers can observe. New tools like laser face plates, which enhance contrast in electron microscopy without increasing damage to cells, could push visibility from a narrow slice of a cellular activity to more than a half of the proteome. Wow. Okay, good to know. I think for today I'm more than done. My brain is almost dead trying to read in English, understand what I'm reading, and not making too much errors. So, I wish you a fantastic weekend. Thank you very much for being here. And see you the next one.
Come on and cry me a river, cry me a river. I cried a river